My name is Michael McQuitty,
My name is Harsha Pamay.
and this is our blast lab board. For gene 1, we decided that it did support our hypothesis because it showed how the specimen is very closely related to the bird section, which tells us that the fossil species is highly related to birds, and it is also a vertebrate. Gene 2 supports our hypothesis because it shows that the fossil is semi-closely related to the common fruit fly. It only shares like one protein with it, and it's probably due to the fossil also having, they're both heterotrophs.
Gene 3 supports that hypothesis because it shows how the specimen is closely related to many different bird species and how it is before the Homo sapiens. It also proves our theory that the species is a vertebrate and has lungs. Gene 4 supports because it shows how a specimen is a vertebrate, it has reptilian relations, and does not have a palatal valve due to it being a land animal with lungs. Our organism cannot survive underwater due to the development of lungs and not having a necessary palatal valve.
Here's our hypothesis. Our fossil is actually below the feathers, but we did have it in the bird region. And through all of our research, we found out that we were actually wrong. It belonged past the feathers. And we have all of our genes, their cladograms here. Gene 1 was actually a chicken called Gallus gallus, and that was the most closely related thing that we got for the fossil. Gene 2 was your common fruit fly. That's whenever we got the one protein that was related. Gene 3 was the zebra finch, which is another species of bird. And our 4th was a Chinese alligator, which showed that it wasn't quite reptilian because it was located in the bird area. And basically what we got out of all these cladograms is our hypothesis was wrong, and it was in fact the feathered bird that didn't have flight. And our only 100% match was in fact the Gallus gallus, which means it was a chicken. This is our gene ranking. For gene 1, we decided that gene 1 was the most useful as the species didn't have that gene, or more of the species that we tested in. For number 2, we decided that gene 3 because there were some organisms that did have the gene. And for 3, we put gene 4 because it didn't really show much significance about the actual fossil itself and where it should be. And for 4, we put gene number 2 because it showed the most closely related common ancestor.
And right here we have 8 of our key vocabulary words. And our Human Genome Project was an international scientific research project that determined whether the sequence of the nucleotide base pairs that make up the DNA. Our genome is a complete set of genes or genetic material present in the cell organism. Our bioinformatics is a science field that develops methods and hardware tools for understanding biological data. Our BLAST, which stands for Basic Local Alignment Search Tool, is a process of calculation that compares primary biological sequence informations. Our cladogram is a branching diagram showing the cladistic relationship. Our ancestor is a lineage that can trace one species all the way back to its ancestor. Our derived characteristics is a trait shared by members of a group in organisms. Our cladistics is a method used to classify animals and plants according to their common characteristics. And this is our works cited.